Cancer, a disease of uncontrolled cell growth, poses a significant threat to human health. At the heart of this complex disease lies the malfunctioning of genes that regulate cell growth and death. Among these genes, the p53 gene stands out as a crucial tumor suppressor, often dubbed the guardian of our genome. The p53 gene produces the p53 protein, a powerful molecule that acts as a cellular gatekeeper, preventing the uncontrolled proliferation of damaged cells. When functioning correctly, the p53 protein acts as a transcription factor, binding to DNA and regulating the expression of genes involved in cell cycle arrest, DNA repair, and programmed cell death or apoptosis. In healthy cells, the p53 protein remains inactive, only to be activated in response to cellular stress signals such as DNA damage, oncogene activation, or hypoxia. Upon activation, the p53 protein initiates a cascade of events that ultimately determine the fate of a damaged cell. If the damage is repairable, p53 halts cell division, allowing time for repair mechanisms to restore cellular integrity. If the damage is irreparable, p53 triggers apoptosis, eliminating the threat of a potentially cancerous cell. However, in many cancer cells, the p53 gene is mutated, or its function is compromised. This loss of p53 function allows damaged cells to evade the body's natural defenses, leading to uncontrolled growth and tumor formation. The crucial role of p53 in preventing cancer is underscored by the fact that mutations in this gene are found in over 50% of all human cancers. The advent of mRNA vaccine technology has revolutionized the field of medicine, offering new hope for treating a wide range of diseases, including cancer. p53 protein mRNA vaccines represent a promising new approach to cancer treatment, harnessing the power of the body's own immune system to target and destroy cancer cells. mRNA vaccines work by introducing a specific mRNA sequence into the body's cells. This mRNA sequence carries the genetic instructions for producing a specific protein, in this case, the p53 protein. Once inside the cell, the mRNA is translated by ribosomes, the cell's protein synthesis machinery, to produce the desired protein. In the context of cancer treatment, p53 protein mRNA vaccines are designed to deliver mRNA encoding for the wild-type, functional p53 protein into cancer cells. This approach aims to restore the function of the p53 protein within tumor cells, reactivating the body's natural defenses against cancer. The process of developing and administering a p53 protein mRNA vaccine involves several steps. First, the mRNA sequence encoding for the p53 protein is synthesized and encapsulated within lipid nanoparticles. These nanoparticles act as delivery vehicles, protecting the mRNA from degradation and facilitating its entry into cells. Once administered to the patient, the nanoparticles carrying the p53 mRNA are taken up by cells, where the mRNA is released and translated into the p53 protein. The newly synthesized p53 protein can then exert its tumor-suppressing effects, inducing cell cycle arrest, DNA repair, or apoptosis in cancer cells. p53 protein mRNA vaccines offer several potential advantages over traditional cancer treatments, such as chemotherapy and radiation therapy. These advantages stem from the targeted nature of mRNA vaccines and their ability to harness the power of the immune system. One of the most significant advantages of p53 protein mRNA vaccines is their specificity. Unlike traditional chemotherapy drugs that target rapidly dividing cells indiscriminately, affecting both cancerous and healthy cells, p53 mRNA vaccines specifically target cells with dysfunctional or absent p53 protein. This targeted approach has the potential to minimize the side effects associated with traditional cancer treatments, improving patients' quality of life during treatment. Another key advantage of p53 protein mRNA vaccines lies in their ability to stimulate a long-lasting immune response against cancer cells. By introducing the p53 protein into the body, these vaccines can trigger the immune system to recognize and target cancer cells expressing abnormal p53. Furthermore, p53 protein mRNA vaccines offer the potential for personalized cancer treatment. The genetic sequence of the p53 gene can vary slightly between individuals, and p53 protein mRNA vaccines can be tailored to target specific p53 mutations present in an individual's tumor. This personalized approach could lead to more effective and targeted cancer therapies. While p53 protein mRNA vaccines hold immense promise for the future of cancer treatment, several challenges and limitations need to be addressed before these therapies can be widely implemented in clinical practice. One of the main challenges lies in optimizing the delivery of mRNA to target cells. While lipid nanoparticles have shown promise as delivery vehicles, further research is needed to improve their stability, biodistribution, and cellular uptake. Efficient delivery of mRNA to tumor cells is crucial for maximizing the efficacy of p53 protein mRNA vaccines. Another challenge is the potential for off-target effects. While p53 protein mRNA vaccines are designed to target cancer cells specifically, there is a possibility that the mRNA could be taken up by healthy cells, leading to unintended consequences. Further research is needed to ensure the safety and specificity of these vaccines. Moreover, the manufacturing and storage of mRNA vaccines can be complex and expensive. mRNA molecules are highly unstable and require specialized storage conditions to maintain their integrity. These factors could limit the accessibility and affordability of p53 protein mRNA vaccines, particularly in resource-limited settings. Despite these challenges, the potential benefits of p53 protein mRNA vaccines in cancer treatment are undeniable. Ongoing research and clinical trials are paving the way for the development of safer, more effective, and personalized cancer therapies that could revolutionize the way we approach cancer treatment in the future.